So now I'm very pleased to welcome Andrew deMello to share with us your works. So Andrew is a professor of biochemical engineering in the Department of Chemistry and Applied Bioscience at ETH Zurich. So it's a very prestigious lab in a very prestigious university. And you are also a co-founder of Molecular Vision, an Imperial College spin-out company developing low-cost diagnostic devices for use in doctor's surgery and at home. And your research interests cover a broad range of activities in, I would say, general area of microfluidics and nanoscale science. So the floor is yours.
Thank you very much. My video doesn't seem to be working, but I hope you can see my presentation. So first of all, I'd like to wish everyone at Fluigent a very happy 15th birthday. As we've heard, the company started from very humble beginnings, and I think it's very clear now that you're one of the leaders in flow control, not just in microfluidics, but in many other areas. So it's lovely that I can be here this evening. What I wanted to do is maybe just give you a very, very quick overview of some of the work that we've done over the past 15 years. So I thought, what were we doing back in 2006, 2007? And what we were doing then was we were using continuous flow systems to make molecules, or in this case here, to make nanomaterials and quantum dots. And at the time, we realized that microfluidics, as we've heard in a number of the talks today and yesterday, really gives you very nice advantage at a really fundamental level, just our ability to control both mass and heat transfer. And we realized, particularly for the synthesis of quantum dots, what was critical was controlling nucleation and growth. And we realized that you could use very, very simple systems and tune both the electronic and optical properties of the material in a very, very easy way. So this is just one example of work that we were doing around that time. And I think it's really a very early example of using machine learning to direct a synthetic process. So what we were doing here, if you look at the little schematic on the top right-hand side, is using a glass continuous flow device. It has a number of inputs. We were using the microfluidic reactor to hopefully make good quality nanomaterials, but then importantly, we used a detection system to extract information about the product on a millisecond timescale. Once we've done that, we then used a control system to basically look at how well we'd done in that experiment, and then to change variables to improve our product. And you can see on the left-hand side there, just an example of a screening run where we could create lots and lots of different nanoparticle populations. And the blue colored dots there are ones which are very close to our target wavelength, which was 530 nanometers. And this really showed that by doing your reaction well and very quickly, if you could marry it with good detection and then machine learning, you could really create a very powerful system. And so we use this kind of microfluidic platform for a number of years, but ultimately, as we all know, that there are significant problems with continuous flows, and these are really stem from the parabolic flow profile that we develop within most micro and nanofluidic systems. And for us, this really proved a problem, particularly for timing reactions. And so we tried to think of different ways to get around this, and we were lucky that at the time, a number of lovely papers from people like yesterday's speaker, Dave Waits, but particularly Rustam Ismaghilov's group at the University of Chicago, started to show how you could maybe use droplets to very good effect in synthetic processes. And so we started to use droplets for a whole variety of processes. I'm sure Dave went through droplets yesterday, so I'll just really highlight a few of the key features, and you'll see in the next few slides that we could really use very, very simple passive microfluidic systems to very, very good advantage, both in performing reactions, but also in performing assays. The key features of droplets for us, particularly for reactions, are the fact that our droplets move at a constant linear velocity. So that means that mass transport will occur without dispersion. it becomes very, very easy to load in different amounts of solute into the droplet, so we can begin to change the chemical payload, again at very, very high speed. And we can make droplets very, very quickly. So it's very easy for us to make very large numbers of monodispersed droplets in very short time. So this kind of experimental platform really is quite seductive when you start to think about doing high throughput chemistry and biology. So we spent, especially in the early years, a lot of time looking at how we might create functional components to do different jobs, things that we would normally do on the benchtop. Could we create devices that would do each of these operations efficiently? And the intervening years has shown by our group and by many other groups that you can very easily do that. So we have devices to mix, to split, to merge, maybe to incubate droplets, and to inject additional reagents into them, and ultimately to sort them as well. So just to show some videos, which are always nice for droplet-based systems, videos that I'm sure you've seen before. You can see here on the left-hand side examples of a simple flow focusing geometry where we're making droplets that vary from a few tens of picoliters up to a nanoliter at rates between 50 and 1,000 hertz. We can make very small droplets very easily, and these are particularly useful for us today when we make particular types of nanomaterial. But you can see here making 75 femtoliter droplets at rates in excess of 20 kilohertz. And then by controlling the rheological properties of the system, we can also create more interesting emulsions, as you can see here. We spend a lot of time, apart from generating droplets, also thinking about how we might process them. And you can see two examples here. The example on the left-hand side is a very old device that we use to merge droplets, in this case, merging two droplets together, but normally between 2 and 6. And here, by really including a pillar array, what we can do is to create a filter for the continuous phase. And that allows us over a period of maybe 30 or 40 milliseconds to have a very robust reproducible mixing event. And we've used this synthetically for a lot of different applications. And one of the key features that we love so much about droplets is the fact that we can keep our reagents, which are in the discrete phase, away from the walls of the microfluidic channel. So this allows us to do things over very long time scales. In the center, you can actually see a very similar structure, again, based on a soft valve, but this is a device that allows us to do dilution or concentration variation, where we have a large mother droplet at the center that you can see here. This has got a volume of maybe around 5 nanoliters. And then as we bring in empty droplets that just contain our buffer or solvent, we can, through a process of merging, mixing, and resplitting, create a daughter downstream droplet where we know the concentration. And importantly for us, we can change concentration again on a millisecond timescale. So we use a bunch of components that we've either created ourselves or we've pinched from many other groups, and we do lots of different things. So just to give two very brief examples here in terms of biology and chemistry. On the left-hand side, you can see an example of one of the early continuous flow, or sorry, droplet-based PCR devices. So this is work in collaboration with Florian Hoffelder's group at the University of Cambridge, where we created a radial device that used the ideas of continuous flow to perform PCR. So the idea there being that we could use the very, very low thermal mass to affect very fast amplification, but through the encapsulation of the reagents into droplets, we were able to really improve efficiencies, ensure that we didn't get contamination in the system, and show very fast and efficient single copy PCR. And I think, as we all know now, this kind of technology has been commercialized in a number of areas, as we've actually heard about today with companies like Stiller Technologies. And I think is one of the nice applications and devices which I think has begun to change the way that the biologists might think about doing experiments. On the right-hand side, you can see here a very, very simple device to make nanomaterials. So in this case, we're using capillary flow reactors to generate a segmented flow, and in this case, create cesium lead halide perovskite nanomaterials. And the beauty of this system here is that by ensuring very, very uniform temperature control, we can, I hope you can see here, really tune the emission of these beautifully emissive materials by controlling the environmental conditions and also controlling the chemistry. So again, here microfluidics really allows you to start making very, very novel materials and perform bespoke material synthesis. Now, however nice droplets are and however nice microfluidic systems are, as we heard from Jean-Louis' talk, detection is always a key issue. And I guess the basic idea here is that as we get smaller, we may be able to improve our chemical or biological process, but as we get smaller, detection becomes progressively harder. So we've spent many years now looking at how we might improve this situation, and particularly with a view to droplet detection. One of the problems we had for many years was really maximizing our ability to extract information. As you've already seen, droplets are wonderful at generating chemical information. We can create droplets at incredibly high frequencies, but that will all be wasted if our detection systems aren't good enough to pull things out. And you can see here at the bottom an example, a beautiful example of work from Rustam Ismaghilov's group, where they showed one of the first kinetic experiments using droplets many years ago now. And through creating identical droplets and performing enzymatic assays within a droplet stream, they could then create these very nice kinetic curves. But one thing that you can see from the image on the left-hand side is that we can't see any droplets. In this case, the droplets are moving far too quickly. So we use, in this case, the average signal to create the kinetic data. And one of our key challenges was, could we now maybe think of ways of starting to look at those droplets, stopping them blur, and extracting much more information from similar systems? And you can see the problem in the image of a London bus on the top of the slide here. So the idea is the foreground and the background are nice and sharp because they're still, but because the bus is moving at a high speed compared to the shutter speed, we can't see it very well. So what we did, and as is often the case, we realized that the answer had been developed many years before, is we really borrowed the ideas pioneered by people like Harold Edgerton at MIT to create these beautiful stroboscopic photographs. And the idea here is that we can freeze a very, very fast event, not by having a short shutter speed, but by illuminating the scene for a very short period of time. And so this is exactly what we started to do over the past 3 or 4 years when we come to look at our droplet systems. And this gives us some very nice advantage. So I don't have a huge amount of time to show you complete studies, but maybe if we look at one of the first things that we did with stroboscopic illumination, we used a standard droplet-based system to perform our enzymatic assay, where we could vary the concentration of our enzyme and substrate. We used little wiggles in the channel to ensure rapid mixing by a chaotic advection. And then we used a CMOS camera to image the droplets as they go through and hopefully monitor the production of a fluorescent product. Now, when we do this and when we synchronize our excitation pulses with our detector, we get a very nice result. So you can see here in the top panel, an image of a droplet flow with normal continuous illumination. We see very high signal, but complete blurring of the droplets, so we can't see them at all. But as soon as we use pulse excitation, we can very nicely freeze the motion of the droplets, and then we can look into individual droplets and extract our information. And this becomes very powerful because we know that we can create lots and lots of droplets per unit time. So you can see here just one example of a very simple Michaelis-Menten analysis here. And the only reason I'm showing you this is that this plot here allows us to extract our Km, which is of very good quality. But importantly, we performed here about 1,500 discrete experiments. These aren't repeats, they're chemically distinct in about 10 seconds. And so really the marriage of our high throughput droplet microfluidics and our good optical detection really allows us to extract lots and lots of information. So moving towards the last thing that I wanted to talk about in terms of droplets is that we can use these systems to good effect to do much more elaborate analyses. So you can see here an example of recent work where we've been using a little bit of a more sophisticated system to look at very fast enzyme reactions. So if you look at the little cartoon at the top, you can see the structure of our microfluidic device. What we do here is we use an acceleration process to mix or to generate our droplets very quickly, and then wiggles to enhance chaotic advection. Hopefully that allows us to mix our reagents over a very fast time scale, and then we slow them down and use stroboscopic illumination to image our droplet train. And by using this approach, again, in terms of the microfluidic device, very simple to make, you can get some very impressive results. So you can see here just some characterization experiments where we can show that the mixing time using this system is sub-milliseconds. So we mix on around a 700-800 microsecond time scale, which means that we can start to probe very, very fast enzymatic processes. You can see here an example of 4 essentially random enzymatic assays. And really the reason I'm showing you these is really for you to look at the time scale. So we can look at kinetic profiles here over maybe 20 nanoseconds, or using an extended device over maybe 400 milliseconds. And that then allows us to really start to look at early time kinetics of a whole range of different enzymatic reactions. In this case, we were particularly interested in dehalogenase enzymes. And again, without going into detail, you can see that the approach allows us to generate lots of kinetic data very, very quickly. And importantly, in this case, allowed us to correlate changes in the substrate binding mechanism with changes in the enzyme structure. So this kind of tool really allows you to generate huge amounts of information, and again, really benefits both the fast microfluidic aspects of droplet-based systems, but also the very fast and efficient detection using stroboscopic illumination. So in the last couple of minutes, I just wanted to show you a couple of examples where we now use the technique of stroboscopic illumination for not just droplets, in this case for imaging flow cytometry. Without going into detail, I'm hoping you can see that we have a very simple microfluidic system or cytometer that consists of a single channel. But by using viscoelastic cell focusing and stroboscopic illumination, we can start to do incredibly high throughput imaging flow cytometry. So here in bright field imaging at rates of around 400,000 cells per second, using fluorescence at rates of around 60 to 70,000 cells per second. And I'm hoping that you can see in these images here, cells moving at very high speed can still be imaged with diffraction limited resolution. And in this case, we can pull out signals from p-bodies and stress granules. So where this is going is in a number of areas, but one area I thought was quite nice to highlight here, where we make use of Fluigent's beautiful pressure pumps, is to look at clinical samples. By using exquisite flow control on small sample volumes, we can inject bodily fluids into our imaging flow cytometer, which you can see in the image there, which is relatively small in size. We can digitize those images and then use machine learning to create diagnostic signatures. And you can see here examples of some of those analyses. This is looking at a particular type of lymphoma, here Sézary syndrome. And by using our imaging flow cytometer and machine learning, we can extract diagnostic results in extremely short periods of time. So my last slide is really to show one other example where we're making very good use of Fluigent's pressure pump technology, here in something very different and very new for us, where we're using valve-based microfluidics. I have 2 small videos here, but hopefully you can see that by using leaky pneumatic valves, what we can do is start to trap single cells or collections of very small numbers of cells in a system. And then by controlling the pneumatics, we can then release the cell, in this case, deliver it to a lysis chamber here. We can lyse the cell and then use high-end mass spec to start to extract proteomic maps from very, very small numbers of cells. So this is an area that we're very excited about, and as I said, makes use of Fluigent's wonderful technology. So really to finish, of course, I must thank the people who did the work, which certainly wasn't me. So these are just members of my current group, and I should acknowledge our other alumni and collaborators. I should also thank our funders and also once again wish all of you at Fluigent a very, very happy birthday.
Are there any questions? Yes.
Do you have some precisions on the single cell proteomics and how deep the analysis can be on single cell?
Can you hear, Andrew?
Yeah, I think I heard that. So that's a really excellent question, one that I'm absolutely not able to answer. So we're very lucky to be working with Rudy Abazol's group here at ETH, and really the innovation here is the fact that in recent years the sensitivity of mass spec detection has become incredibly powerful. And so it's allowing us through very good fluid control to be able to really take those single cells and deliver them. But I think, as you could have seen, I've stopped sharing now, but in the data that I showed, that we can extract signatures from a single cell of well over 1,500 proteins, and that scales in a very interesting way. So I think we're really at the beginning of that journey, but I think the key challenge here will be using data analysis to really pull out the richness and complexity of the signal.
Okay, there is one online question. So have you experienced any droplet coalescence in your experiments? How have you overcome that? And if so, what surfactants did you use?
Oh, I'm sure that everyone's laughing who's ever played with droplet-based systems. So of course, coalescence is really one of what I would call the elephants in the room. So one of the problems that we have to deal with every day. Obviously, surfactants, particularly perfluorinated surfactants, have been proved to be wonderfully useful. It's very, very interesting. I mean, there have been some really good studies by people like Jean-Christophe Barret who've really started to look at these. It is an area that I think as a field, we've actually done a very bad job at really starting to understand these. So to answer the question, we use surfactants. Surfactants normally do a very good job for us, particularly over relatively short timescales. So I think droplet systems work very well over the time scale of the experiments, and they're typically short. I think the real challenge becomes when you want to store droplets over long periods of time. To do that, you're typically going to have them in close proximity with very little oil surrounding them. And that's going to lead to more problems. So I think to give you the honest answer, surfactants allow us to deal with coalescence very, very well, but there's still a lot of work to be done.
Which one? Okay, thank you. Yes, there is another one.
I have a question on the imaging flow cytometry. So you talked about the incredible rates of making images of the cells in your device. Can you also sort cells in your device? And if so, how much of this speed do you have to reduce to actually being able to sort?
These are great questions. So this is work that we're starting to look at now. That's a lot more challenging. So in terms of flow cytometry, what's really nice here is that we can use sheathless systems. So by using viscoelastic and lift forces, we can ensure that our cells are moving where we want them. To start to do sorting at those kinds of rates is going to be incredibly difficult. And so I think that that's an area that we'll have to start working with. I should say actually that although imaging flow cytometry is very nice, we're getting particularly interested in using microfluidic flows to generate deformation, so using deformability as a phenotype. And in those systems, I think the throughputs that we would work with would be slightly less. And so we could start to look at sorting. I mean, sorting is a big issue, not just in, say, flow cytometry, but certainly with the droplet-based systems that I was talking about. We would typically sort at rates, if I'm being completely honest with you, no more than about 2,500 or 3,000 droplets per second, which is great, but it still doesn't match with the throughput of the droplets. And similarly for the imaging flow cytometer, our throughput in terms of imaging will always be much, much higher. So again, I haven't given you a proper answer there, but that's definitely a work in progress.
Thank you.
Thank you. And maybe a last one. Have you had non-specific binding of cells to microchannel? How to compensate the background?
So typically the work that we do with cells, apart from the last example there, we encapsulate cells in droplets. So whether they're small numbers of cells or down at limiting dilution single cells, we keep them within the droplet. So we try and avoid that. And again, I think more generally, if I was being honest with you, I said that one of the reasons that we started to use droplet-based system was driven by controlling residence time distributions. That's true to some extent, but I think, again, from a practical point of view, using droplets really starts to remove exactly those kinds of problems. We keep material away from the surface, whether it be small molecules or nanomaterials or cells or DNA. I mean, one of the reasons that we worked a lot on developing droplet-based PCR was that when we initially showed continuous flow PCR, it worked, but it worked at, under very, very unusual conditions where we had to use very high concentrations of polymerase. And this is exactly down to the fact that the surface area to volume ratios are so high that molecules, cells, and DNA will stick to them. So we tend to try and keep the surface passive and to try and remove those kinds of features. But clearly in the last example that I showed you there, surface interactions do become very important.
Okay, thank you very much again.